Hello, everyone, and welcome to this webinar on design considerations for bioequivalence studies, which is the latest in our monthly webinar series. In this webinar, we will give an overview of key concepts of bioequivalence studies, an overview of commonly used crossover designs in bioequivalence studies, and we'll look at some examples of each of these as well. In nQuery. But before we get too deep into that, I'm just going to run through some quick bits of housekeeping. So today's webinar is being recorded, and the recording, along with the slides used and the example files, will be sent to you shortly after we conclude here today. If you have any questions, please feel free to type those in the chat box, and I'll do my best to leave some time for those at the end. However, if we do run into any time constraints, I'll make sure to reply to all of those questions by email after the webinar as soon as I possibly can. Also, if you think of a question afterwards and you send it in after the webinar is finished, I can respond to those by email as well. You can also use the chat box to inform us if you are dealing with any technical difficulties, and one of my colleagues will do their best to resolve that for you as quickly as possible. So, by way of a quick introduction, my name is Denis Desmond. I'm a research statistician here at StatSolves, and I'm going to be your host for this webinar that is brought to you by nQuery. And yeah, massive thank you to all of you for taking the time to join us here today. So as I briefly alluded to, just a quick rundown of the agenda for today. Firstly, we're going to start off with an introduction to bioequivalence, which is the topic for discussion today. We're then going to go through an overview for some crossover designs, look at some advantages, disadvantages, things like that. Next, we'll look at some examples of crossover trials and sample size determination in nQuery. And then finally, we'll wrap things up with discussion, conclusions, and the questions as well, if time allows. So, as I mentioned, this webinar is brought to you by nQuery, which is the complete solution for optimizing your clinical trial designs. nQuery is very widely used in the pharmaceutical and biotechnology industries and offers sample size determination and power calculations from preclinical research through to post-marketing studies, with the main focus being on confirmatory phase 3 trials. And nQuery features over 1,000 scenarios for fixed-term Bayesian and adaptive studies. If you're interested in learning more, you can see the resource section of our website for demos, work examples, previous webinars, and future webinars will be advertised on there as well. So nQuery operates on a tiered licensing model. So each of these 4 tiers gives you access to different feature sets depending on the tier that you purchase. And it's a progressive model, so if you buy or upgrade to a higher tier, then you will continue to have access to all the features from the previous tiers as well. So the basic tier is the tier available to all customers who purchase an nQuery license, and this focuses on sample size and power for a variety of designs, endpoints, and hypotheses. The Plus tier focuses on our Bayesian features such as credible intervals and assurance. And the Pro tier, which is our most popular tier, includes adaptive design features such as group sequential designs and multi-arm, multi-stage designs. And then lastly, there is Expert tier, which currently focuses on simulation tools for milestone predictions for enrollment and event milestones. So if you'd like any more details on this, you can follow the URL at the bottom of the slide here once they're sent out to you. And that will show you the package comparison tool that we have on the website, giving you detailed breakdown as well of the tier required for each table that's offered in nQuery. On that as well, if you don't already have a license of nQuery or you'd like to try a higher tier, you can get a full trial of the full features of nQuery from our website, but I will go through that a bit more in the end. So I think that is all the kind of housekeeping bits covered. So with that, yeah, we're going to jump into the first section of today's webinar, which is an introduction to bioequivalence studies. Very quickly first though, what I'll do is just give a quick bit of context, kind of set the scene, if you will, just regarding clinical trials and different hypothesis types. Obviously that's not the focus for today. And so if you'd like to learn more about different hypotheses, you can find a previous webinar that we did on that topic on our website as well. But just very quickly, sorry, excuse me. In clinical trials, the objective is usually to evaluate efficacy and safety of a new treatment, and a hypothesis is formed based on these primary study objectives. And so a hypothesis statement is a statement made about the effectiveness or the safety of this new treatment in the population of interest, and it defines the study variables that are considered the primary clinical endpoint, so that hypothesis can be tested then using an appropriate statistical method. So, for example, in the typical inequality testing scenario, an example of a hypothesis might be that there is a treatment effect, but the definition of efficacy will depend on the clinical and regulatory scenarios. So that might be, a new treatment approval or an approval of a generic equivalent to a pre-existing treatment. And so today we're obviously going to be focusing on those equivalence type hypotheses, but obviously it's also of interest in certain scenarios to see if a treatment is non-inferior to or superior to a standard treatment as well. So in equivalence testing, you are testing if the new treatment is equivalent to the control or reference, which is going to be the current standard treatment. And this test is carried out using an indirect measure without a good or bad direction necessarily. So direct measure is something like mortality or blood pressure, where there's like an obvious good direction. But indirect measures are something we're going to go into more detail in a minute, but essentially, there'll be things like the concentration of the blood concentration of the drug in the bloodstream, over time. And so there's no necessarily good direction for that. Instead, you want it to fall within lower and upper margins of equivalence that have been set by regulators. So this testing procedure for equivalence tests is known as the two one-sided tests, or TOST approach. So this uses a null hypothesis that is split between the difference between the two treatments is greater than the upper limit, or the difference between the two treatments is lower is less than the lower limit. And then the alternative hypothesis being that the difference falls within these limits. And so each of these null hypotheses are tested at the one-sided significance level, and the overall type 1 error is equal to the one-sided alpha for each of these tests without the need for multiplicity adjustments. But if you want to use instead a confidence interval method, you need to use twice the TOST alpha, because it's not a two-sided test. So for example, a 5% TOST alpha would mean a 90% confidence interval. Okay, so now that we've covered that in the intro, we can move on to looking at bioequivalence itself in more detail. So first of all, a definition per the ICH guidelines. Bioequivalence is defined as two drug products containing the same drug or drugs are considered to be bioequivalent if their relative bioavailability, which refers to the rate and extent of the drug absorption after administration in the same molar dose, lies within acceptable predefined limits. So the exact wording of the definition of bioequivalence and bioavailability will change a little bit across different agencies. But just to break that down a little bit, if two products that have the same active ingredient administered in the same quantities display similar biological properties, then it can be assumed that they have an equivalent effect. So, where is this useful? Well, first of all, in the area of generic drug development. So when a new treatment, sometimes referred to as an innovator drug, has gone through all the various clinical trials phases and receives its regulatory approval, it can become known as what's called a reference listed drug. And this company can, patent that drug, and that patent will eventually expire. And so as that happens, other companies can then make their versions of this drug. But rather than having to repeat the whole clinical trial process from scratch, as if it were a new treatment to show safety and efficacy, companies can submit abbreviated applications showing that the off-brand version is equivalent in terms of bioavailability to the reference listed drug that has already gone through that process. So like if we just skip ahead for 1 second to the next point on the slide, demonstrating bioequivalence is an accepted surrogate for demonstrating therapeutic equivalence. So in other words, if the new generic is equivalent to the reference listed drugs in terms of bioavailability, then this key assumption of bioequivalent studies is that they're also equivalent in terms of safety and efficacy. And yeah, like I said, that's the fundamental equivalent for a bioequivalent study. So just on that then, because the aim is to assess the difference in bioavailability between the two products, the bioequivalent studies will mostly be carried out in healthy volunteers in order to minimize that variability. But of course, there'll be exceptions to that as well. So the main one would be if there are any ethical concerns with doing so. So for example, if there are safety concerns around adverse events, that may be an acceptable trade-off in the treatment of like a very chronic illness. That trade-off might not be acceptable in order to administer the treatment to healthy subjects in order to test the comparative bioavailability. So if we go back up to the second point here as well, so the second part or second area where bioequivalence studies are useful is in formulation changes within the innovator drug itself. And so this isn't with respect to the amount of active ingredient available or dose or anything like that. That does need to stay fixed, but it might be, say, switching from 2 5 milligram tablets to 1 10 milligram tablet, or maybe change to some of the non-active ingredients in the formulation either, in order to make sure that changing those non-active ingredients hasn't any knock-on effect in terms of the bioavailability of the formulation. And so the goal of a successful bioequivalence study is to reduce the time and development costs of these generic alternatives by removing the need to do the entire clinical trial process again from scratch, so that these drugs will be more affordable and then also become more widely available for patients. One small note here, actually, I have been saying bioequivalence studies, and it's here in the slides as well. Technically, bioequivalence is the end result of a comparative bioavailability study, but just the term bioequivalence study has become so widely used, and it's used in a lot of regulatory guidance. It's just kind of a convention at this point. So minor point, but just wanted to acknowledge it there quickly. So next we will look at kind of approaches for demonstrating bioequivalence. So there are pharmacokinetic measures, also called PK measures, pharmacodynamic measures that are also called PD measures, and there's comparative clinical trials and in vitro studies. So starting off with pharmacokinetic measures, because these are the preferred measures to be used by regulators such as the FDA or the EMA for in vivo bioequivalence studies. And so in vivo studies cover drugs that are intended to be absorbed into the bloodstream. So if you look over on the right hand side of the slide here, you can see that we've a little graph here that is the concentration time curve. And so on the y-axis, you have the concentration of the drug, and on the x-axis, you have the time. And so this curve is used to analyze the rate of absorption and elimination of the drug of interest in the blood or plasma. So this curve can be generated from taking samples from the subject before administration and then at certain times afterwards. And different agencies will have different requirements for the timing and number of samples that need to be taken. Typically these relate to the half-life of the drug, and you need to take certain samples at certain times in order to accurately predict the concentration time curve. So if you look here, you can see that the area under the curve is also indicated on this graph here, and this is one of the primary measurements of interest in a bioequivalence study. And so the graph here will obviously show AUC from time 0 to time t, whatever the last sampling time is. But for analysis, it's actually the AUC from time 0 to time infinity that needs to be stated. And so typically this first portion of the graph of the AUC up until time t can be calculated with the trapezoidal method, and then the portion from the final time to infinity can be estimated using the elimination rate, of the drug in the body. And so that's why the timing of the sampling is so important as well, because we need to get not only an estimate of the concentration time curve, but also that elimination rate as well. So other measures that are of interest, sometimes these can also be primary measures, but most of the time they'll be secondary measures to the AUC. And so these are the Cmax, which is the maximum concentration of the drug in the body, and you can see the two Cmaxes labeled here. And then Tmax, which if you track down, is the corresponding time at which Cmax occurs. And so, as I said, normally these are secondary interest, but depending on the treatment, these might have more of an impact, and so they might become the primary analysis, and as AUC might become the secondary analysis. So that's pharmacokinetic measures, and they obviously are related to what the body does to the drug, and then pharmacodynamic measures, on the other hand, are measures of what the drug does to the body. So if for some reason, the AUC or other PK measures aren't suitable for a particular product, maybe that's that they're not accurate enough, or but sorry, by accurate I mean it's like they're not easy to be accurately assessed, then you can use pharmacodynamic endpoints, and these relate to relevant physiological changes caused by the treatment in the body. So for example, that might be the impact on blood pressure, and so often these will be used in combination with pharmacokinetic measures rather than as the primary analysis. But again, as I mentioned, some cases necessitate that these will be used instead of PK measures. The third way for approaching the demonstration of bioequivalence is using a comparative clinical trial. And so again, if PK or PD measures aren't suitable, it may be necessary to use a 3-arm trial where the new test treatment is compared to the reference and a control placebo. And then lastly, there are in vitro measures, and these are for products that are not intended to be absorbed into the bloodstream. So, nasal sprays or creams or things like that. And so these measures will be specific to the rate and extent of the active ingredient at the local site of action. So as well as the different approaches, there's also different criterion for assessing bioequivalence, and typically regulators are only going to require evidence for average bioequivalence in order to grant approval for a generic product. And so average bioequivalence is fairly straightforward. It looks to test whether the difference of the averages of the pharmacokinetic parameters like the AUC for the reference and the generic treatment fall within the limits of equivalence. But the main drawback of average bioequivalence is that it doesn't cover drug interchangeability. And so there are two components to drug interchangeability. The first is prescribability. And so this is, the ability of a physician or a doctor to be able to prescribe one of the possible treatments to a new patient, that has not yet started treatment. And then the second part is switchability. And this is relating to the ability of a patient who's already in treatment to switch from one treatment to another. And so because any new generic is going to be assessed for comparative bioavailability compared to a treatment, this isn't as big a concern when switching between the reference listed drug and any one generic. But this becomes very important for switching from one generic to another, and so it has to be certain that these new generics are interchangeable with previous ones. So population bioequivalence is what is used to examine prescribability, and population bioequivalence is examined by comparing the variability of the PK measures between the subjects, and population bioequivalence is demonstrated if the difference lies between the predefined margins set by the regulator. And then individual bioequivalence assesses switchability, and this is again done by comparing the variability of the PK measures, but within the same subject. And like before, individual bioequivalence will be demonstrated if the difference lies within predefined margins. And so on predefined margins, if we move on to the last point here, we can see that these margins will be typically set by the regulator, and the common standard for the geometric mean ratio of the AUC or whatever parameter is being used is that it lies within 0.8 to 1.25. But there are certain scenarios in which, in consultation with regulatory agencies, that limits may be altered based on expertise or the variance. So an example of that would be what are called highly variable drugs, and these are drugs with coefficients of variability of greater than 30%. And it may be possible to widen margins for those drugs, again in consultation with the relevant regulatory agency. And so the reason why these highly variable drugs are tolerated is because they'll have a very wide therapeutic effect. So that means that changes in the concentration of the drug in the body will have a minimal impact on the safety and efficacy. But then on the other hand, if you have drugs that are very narrow therapeutic index drugs, these margins may have to be narrowed or scaled down. And so like a common one for that would be 0.9 to 1.11. And again here, this is set so that the limits are the reciprocal of each other. And so narrow therapeutic index drugs, converse to the highly variable drugs, are extremely sensitive to small changes in concentration. So, efficacy can be dramatically reduced if the concentration is just a small bit too low, and there can be a substantial increase in the risk of adverse or toxic events if the concentration is just a little bit too high. And so, there needs to be a smaller bound when assessing the comparative bioavailability of these two treatments. So now we're going to move on to the next section, which is on crossover designs. And we'll start off again with an introduction. And so a crossover trial is one in which the subjects are given sequences of treatments with the object of studying the difference between individual treatments. And so the simplest and most commonly known and used design is the two-sequence, two-period design, known as the 2x2 design. And so in this treatment, half of the subjects will receive the test drug first and then switch or cross over to the reference drug, and then vice versa for the other half of the subjects. And so you can see here, just down the bottom of the slide, just a diagram of how that would work. And so just the extra point to note is that, subjects will come in and be assessed for eligibility. Once they're enrolled into the study, they'll be randomized into one of the two sequences in this case. And typically this will be double blind, but it is possible, again, in certain instances for it to be open label trial. But just take, for example, sequence 1 here, and they'll start off in the reference treatment in period 1, and then there'll be this washout period before starting the second period, in which they'll take on the test treatment. So the washout period here is very important, and it is a period of rest between the treatments in order for the subject to return to baseline conditions. And this is done so that the effects of the first treatment that the subject undergoes aren't carried over into the second. And so these carryover effects are a very important decision in the consideration of using a crossover design. Obviously, it's not something that you encounter in a parallel design, and regulators will require, a sufficient length of time period for the washout phase. And again, this will be related to the half-life of the drug. And then another aspect of a crossover trial that you don't get with a parallel one, and it's one of the main advantages of choosing crossover design, is that each subject will serve as their own control. And so this allows for within subject comparisons, and it removes the between subject variability from comparisons. And so within subject variability can also be referred to as intra-subject variability. And so measures of, or types of this, examples of this type of variability, can be observed from, maybe biological variability, even though it's in the same subject, or measurement error, in taking samples, such as, measuring the AUC, or rounding after a log transformation or something like that. But the thing with this type of variability is that the intra-subject variability will tend to cancel out in the long run. However, between subject variability, need not necessarily cancel out in the long run. So if we jump onto the next slide here, we can look at that as part of the advantages and disadvantages of crossover designs. And so, we just covered there the reduction in variance. And, this is one of the key points that make crossover design so appealing for bioequivalence studies. Another benefit of the reduced variance is that the sample size required for a crossover design is typically going to be less than a comparable parallel design. With the reduced variance, it's going to lead to more precision in the estimates, and so I think it makes intuitive sense that then that will lead to a reduction in sample size, and so this can lead to some efficiencies in recruitment and save on some costs as well, just from the enrollment side of things. The final point here, not necessarily as relevant for bioequivalence studies, as oftentimes they're conducted in healthy volunteers. Mentioned that earlier, but it is an advantage of crossover designs that in the context where patients are enrolled, that everyone is going to receive the new treatment. So particularly in a trial for an innovator drug, a new treatment that shows good prospects, there can be ethical concerns around randomizing patients to placebo arms for lengthy confirmatory trials, and so this is less of a concern in a crossover trial where in at least one period the subject will receive the treatment. But then we move over to the disadvantages, and Crossover trials are not suitable for all scenarios. An example of which is they're not well suited to illnesses that wouldn't be stable throughout the study period. And they're not suited to treatments that look to have a permanent effect. So that last point there should be kind of, again, fairly self-explanatory. There'd be no point, crossing over to examine, the next treatment if the, illness has been cured in the first period. So as such, crossover designs are, best suited to treatments for ongoing or chronic illnesses where, the goal is, managing symptoms rather than trying to cure an underlying cause. So for example, things like asthma or hypertension, excuse me. So another disadvantage is that there is still potential for carryover effect to be present even without a washout or even with the washout period. So typically drugs will have a single period carryover effect, which is where, it's possible for subjects to return to baseline during the washout period. However, some treatments, could have extremely long half-lives, and as such, they can have carryover effects lasting multiple periods, and these are known as K-order carryover effects. But with that said, we are going to talk about ways of possibly mitigating that in a few moments. And then lastly, there's a higher risk of dropout in crossover studies for two main reasons. The first is that there's more likely to be a longer follow-up time than there would be in a parallel trial. Just because even in like a simple two treatment, two sequence, two period case, subjects are going to be followed for two treatment periods and a washout period in between them. And so this total study time, may be longer than a single intervention during a parallel trial. And the second part of it is, in most cases, there's going to be more patient involvement as well. We mentioned earlier when we were talking about the pharmacokinetic measures that there needs to be a number of samples taken in order to, build a concentration time curve. And so, these there's going to be multiple samples taken over, a number of visits in order to model that for each treatment. And so, especially for multiple or for more than two treatments, this will represent a significant patient involvement. And then the other disadvantage just around dropout itself is that because each subject serves as their own control, the statistical analysis gets much more complicated with dropouts. So oftentimes it's easier to kind of recruit a couple of extra subjects in anticipation of dropout, and just reducing your sample size, but still having sufficient power, rather than trying to do this extra complication. So now we are going to come to higher order crossover designs. And so higher order designs are those that have greater than two sequences, greater than two periods, or both. And so the notation kind of commonly used for these is treatments times sequences times periods. And so in the 2x2 design, each subject only receives each treatment once. But if we add replications to the design, so replications of sequences, the intra-subject variation can be used to estimate both the direct drug effect and also the carryover effect. And so, that makes it very appealing, like I said, for, use with drugs where maybe the K over or K order carryover effects are expected, because these designs can be used in the presence of a carryover effect. Higher order designs can be used for average and population bioequivalence as well, and in many cases they'll be strongly recommended depending on the treatments, but they are required for assessing individual bioequivalence, because when we talked earlier about switchability, that's a within subject assessment, and so in the 2x2 design there's no replication, so it's not possible in that study design to get unbiased estimates for the intra-subject variability, and so it's just not suitable at all for use of bioequivalence or individual bioequivalence. So we'll talk about in a minute, common design used for that. Fed versus fasted comparisons then as well. Another common use of higher order crossover designs. This time, it will be, more than one treatment, or more than two treatments, excuse me, sorry. So, when you want to assess the effect of food on the bioavailability, there'd be 4 interventions. You'd have the reference drugs with and without food, and then the test drug with and without food. And then we mentioned earlier the highly variable drugs and the narrow therapeutic index drugs. So when assessing comparative bioavailability for these types of products, the FDA will also require the use of higher order designs. So common designs for higher order crossover with 2 treatments are the 2x4x2, which is also called Balam's design. So this is a fully replicated modification of the 2x2 design. So it adds 2 additional sequences, which is a test-test sequence and a reference-reference sequence. And so that's a very common design used for 2 treatments. Another one is the 2x2x3, which is also called the dual balance design. And so this is balanced in the sense that each treatment follows itself and the other the same number of times. And so again, depending on the context being used, that can be a very desirable property. So then the 2x2x4 design is the one that's recommended for studying individual bioequivalence. And again, this is a fully replicate design. And then lastly, you've got a 2x4x4, which is another fully replicate design that is actually constructed from 2 Balam's designs. And so all of these designs are optimal in the presence of carryover effect. So then there's also designs that will allow for more than 2 treatments. As I mentioned, really the limit is more of a practical one, because once you kind of get over 4 treatments, it's less common to see those types of designs, just purely because costs will increase, and then the demands on the subjects are going to increase as well, as you study more treatments and more periods, and then there's going to be lots and lots of samples and a much longer follow-up, the more periods that are added. But for the design of these, higher order crossover trials, Latin squares are used for greater than 2 treatments. And these have the nice property that every treatment will have the same frequency in each period. So if, for example, we look at this 3x3 design here, or 3x3x3, I should say, sorry. So each of these represents a sequence, and then in each sequence, it's indicating which treatment is being administered in each period. And so, with this design, you can see here that, treatment A, B, and C are all being assessed in period 1. Treatment A, B, and C are all being assessed in period 2, and then treatment A, B, and C are all being assessed in period 3. And so, again, there's certain instances, like an example with carryover effects, where this is a desired property. And then Williams squares are kind of a subset or a special type of Latin square, with the additional property that each treatment follows every other treatment the same number of times. So here, if we look at the 4x4x4 Williams design, obviously, same as above, in each period, each treatment is studied the same number of times, so A, B, C, D. You don't need to go through that all again. But what we can show is as well that, like, B will follow A once, B will follow D once, and B will follow C once. And then, A will follow D once, A will follow C once, and A will follow B once. And so again, this is considered to be an optimal design, and again, well suited to testing carryover effects. So now that we've kind of covered the theory, we're going to move over and look at some practical examples in nQuery. So our first example today is from a two-way crossover study between test and reference drug. And on the left here, we have a snippet from that paper, and that details that the pharmacokinetic parameters used were Cmax and AUC, and the highest coefficient of variation for those was estimated to be at 0.383. The significance level of the test was 5%, and the expected test reference ratio was 1, and that the limits for equivalence were 80 to 125%. And so with all that information, they deemed that 50 subjects would be considered sufficient to attain the target power of 80%. And so we've just that summarized in a table here as well on the right. And with that, we can switch over to nQuery. So here I have open the table used for equivalence testing for crossover designs on the log scale ratio, MT40. And so for those of you that may not be as familiar with nQuery, this is how a lot of the tables will look. And the left-hand side here, you have the row names, and they will indicate what values to be input in each of the little cells here. Each of the columns will consist of a calculation, and then the yellow rows that are highlighted here will indicate rows that can be solved for. below the table here, we have what we call a side table. And so side tables will sometimes be used if you need to provide more information than just a single value into a cell for a main table row. In this case, this is an optional side table, and it just gives you the option to specify either the standard error or the mean squared error. That will then convert that to the coefficient of variance and transfer it to the main table. On the right-hand side here, then you've got the help cards, which will give you some additional information and guidance about potential row values for each of the rows here, and the home card as well, just detailing a little bit extra about the table itself and the design. So yeah, that's the basics anyway, and enough to kind of follow along with the example. So with that, we'll dig in, and we'll start by filling in 0.05. Now, the study design here, this is a drop-down row, and so 10 in total, but it gives you the option to choose a number of different higher order designs. But as this example is 2 by 2, we'll just leave it at the default, and then we will fill in 0.8, 1.25. The expected ratio was 1, and the coefficient of variation was 0.383. So these next two rows are drop-down rows as well. So the power calculation method row, here you can see there's 4 options, and this just relates to the method used in the calculation of the power. There's 2 exact methods, one evaluated using the Owens Q approach, and the other evaluated using direct integration of the non-central t distribution. And then there's also 2 approximate approaches as well, one based on the non-central t and one based on the shifted t. We're going to leave this at the default as well. Unless you have specific reasons for doing so, there's no reason not to use an exact method. And then the reason, that you might choose the Owens Q over the direct integration is just purely that if sample sizes were to get very large, that there's a slight efficiency to be gained in using Owens Q. The robust degrees of freedom dropdown is a simple yes, no. This doesn't apply here, but what it is it's the robust degrees of freedom, or the degrees of freedom suggested by the Sen basic estimator approach. And this is applicable from some higher order designs, and it's simply calculated as n minus sequences. And there can be certain situations for some of the replicate designs where, depending on the model used, it might be more appropriate to use the robust degrees of freedom. But like I said, for this case, it's not applicable, so we'll just leave it at no. And then last thing that we want to do is that we will enter in our target sample size of 50, and you can see here that the target power is met. So with that, then we can go back to the slides here, and we can go to our second example. And so this is just a toy example using data from the BBAC website, which is a really great resource for bioequivalence and crossover designs. It's included in the reference at the end of the slides. And what they've done here is they've set up a 3 treatment, 6 sequence, 3 period Williams design. They assume a test reference ratio of 0.95, coefficient of variation of 0.25, and then the usual 0.05 alpha significance level, and the bioequivalence limits of 0.8 to 1.25. And so just we can see here from the tabular summary that they're saying that the sample size of 30 will be sufficient to reach the target power. So if we move back over to nQuery, just to keep things clear, I'll move over to a fresh version of MT40 and start here filling in 0.05. This time we're going to click the drop down and select the 3x6x3 Williams design. Again, fill in the same bioequivalence limits. This time the true ratio is expected to be 0.95 and the coefficient of variation 0.25. Again, we'll keep these at the defaults and we'll plug in 80%. So the thing here is that we get 27, not 30. And so if we just click over to the output statement here for a second, one of the things with this table is that it will solve for the minimum sample size required to meet the target power. So in this case, you can see that this is an unbalanced design. So 3 of the sequences will have 5 subjects and 3 of the sequences will have 4 subjects. And so if I just copy and paste in here, and we instead solve for power with 30 subjects, we'll see that we still clear the target sample size and we get our balanced sequence with 5 subjects in each. And so this doesn't mean that you have to use an unbalanced design. Again, it's just showing you how many subjects are required to meet your target power. And so because 27 isn't a multiple of the number of sequences, it's going to be an unbalanced design. So if you go to the next multiple of your sequences, that will give you the balanced design and also give you that little bit of leeway for dropout as well, while still having sufficient power. Okay, and so we go back again to the slides and we'll look at another one final example. And so again, this is from BBAC, and up to now we've been dealing with the standard limits for bioequivalence, but let's just assume in this case that we're dealing with a narrow therapeutic index drug, and so instead we're going to have to use the stricter limits suggested by the regulators of 0.9 and 1.1111. So here we're going to use the 2-sequence 4-period replicant design, and there's going to be a coefficient of variation of 0.125, an assumed test reference ratio of 0.95, and again we'll target a power of 80%. So we'll swap back, and again, just for neatness, we'll move over to a new version of MT40. Again, we'll fill in 0.05, and this time we're going to want 2 by 2 by 4, 0.9, 1.1111, 0.95, 125, and again we'll keep these at the default, and we're going to look for 80% power. And so here we get 34, which just needs to be done, but confirming with the output statement, it's a balanced design already because it's a multiple of the sequences. And so the only thing that we'll just do before we finish up, just because I'm conscious that we're running tight on time, is just, again, we mentioned earlier that it would be intuitively obvious, but if instead we set this back to the kind of regularly used bioequivalence limits, you can see that there's a dramatic reduction in the sample size required. And so, that makes sense, of course, like, the narrower band around the treatments will require a higher sample size in order to show that. And so, yeah, we'll just switch back to the slides there now, and we'll just finish up there now. Just again, conscious of time now, sorry about that. So, bioequivalence studies we saw today are those which aim to demonstrate equivalence with respect to bioavailability as a surrogate for therapeutic equivalence. And so this is important because doing so, instead of having to, treat a generic drug as if it was a new treatment, reduces the costs associated with the clinical trial, and they can be kept down, and the process can be completed sooner. And so treatments are more affordable and more easily available for patients. I mentioned as well, bioequivalence studies are crucial in the fields of generic medicines, and the most common type of carryover or crossover design, is the 2x2. But crossover designs are used because they offer many potential benefits in bioequivalence studies. And so the biggest advantage is that each subject will serve as their own control. And so the between subject variability can be eliminated. And, as well, certain designs, replicate designs can facilitate unbiased estimates for the intra-subject variability. And so this reduced variability can lead to sample size efficiencies when compared to parallel trials. Lastly then, higher order crossover designs can be very useful if carryover is a concern, even with included washout periods. And this helps to minimize one of the main potential drawbacks when considering a 2x2 crossover design. But not only that, they're also crucial in other areas, such as individual bioequivalence, and also the assessment of highly variable drugs and narrow therapeutic index drugs. So, yeah, I'm very sorry, but I am after running over time here, so I am going to have to leave the questions for now, unfortunately. But like I said, I will get back to them via email as soon as I possibly can. And apologies again for that. Just to cover the last few bits here before we finish up, if you don't already have a license for nQuery and you'd like to try it out, or play with any of the examples that we saw here today, or you have nQuery but you'd like to try some features from a tier that you don't currently have access to, you can go to the website here, statsols.com/nQuery-demo, and you can get a free 14-day trial with no download or install required. It'll be accessed via browser. There'll be no credit card requirements. It'll be just a provision of an email address. And so this trial will give you access to the newest version of the software and all of the tiers. So you'll be able to try out, all the tables, for fixed term adaptive designs and milestone prediction as well. So yeah, like any additional questions, you can email them in. The questions that have come in, I'll get back to them as quick as I can. So it's info@statsols.com. If you'd like to contact us, and we'll get back to you as soon as we can. If you have any feedback, we'd love to hear it. If there's an area you'd like to, receive more detail on, just give a shout, or perhaps there's something that you'd like to see featured in a future webinar. And then speaking of which, we have additional content, and that includes the full, library of past webinars, available on our website at statsols.com. And then also at the end of the slides here, you're going to receive these in a minute. You have the references that were used to, put today's webinar together. And again, any questions, just get in touch with us. But with that, I think that I've covered everything today. And just to remind you that we host a new webinar every month, and we hope that you'll join us again soon. So thanks again, and until next time, all the best. All right, best luck. Thank you.